In this video, we're going to pick up where we left off, and we will describe the arterial and venous ends of a capillary. We're going to answer this question. If 5 liters of blood moves through the arterial end of capillaries in 1 minute, how many liters are returned through the venous end of capillaries in 1 minute? Describe the blood flow, including velocity. We'll look at blood pressure, and this really is going to be probably the most interesting or the most critical portion right over here. Okay, so let's go back, and we've seen this picture already. If you look at this picture, you know that I've got the the arteriole and the venial, and the stuff in between here then is your capillary, of course with the medarterial running through. So this is your capillary. If you look at this, what you'll see is that it goes from bright red to kind of a pale blue, or let's just say blue, and that's because you're dumping off oxygen here, as well as other good stuff, as well as picking up waste. And so we're just going to say that, well, this kind of the red part of the capillary versus the blue part of the capillary, we're going to say this is the arterial end of the capillary and this is the venous end of the capillary, but it's still part of the capillary. So pretty simple, pretty straightforward. Okay, the next thing that I want to look at is I want to show you this picture. So what we're going to look here is talk about flow rate, the flow rate. And so what I have here, let's just imagine this is a creek, and then as you go down this creek at point A, and if you were to put a little barrier here and measure stuff, you'd say, oh, there's about 100 gallons of water that passes through per minute. Okay, so this is the flow rate, about 100 gallons of water per minute. At this point, the creek widens, and here the question is, if you place this here to also measure what is the flow rate here, what would your answer be? And your answer should be, well, it should be 100 gallons per minute. Because if it is less than 100 gallons per minute, where's the rest of that water going? It's going to run off onto the banks of this river, okay? And you're going to see flooding. So if you don't want, if this is like a regular non-flooding creek or river, then you're going to see 100 gallons per minute here. And when you get down here, when the creek narrows again, once again, it's still 100 gallons per minute. So again, keep in mind, if this were actually like, let's say, 1,000 gallons per minute, that wouldn't really make much sense because eventually then this whole thing would run dry, right? So the river upstream would run dry. If we said, on the other hand, this thing is 50 or let's say 50 gallons per minute, then again, all that excess water is you're going to see that excess water then overflow, and it's going to end up kind of dumping over here and kind of flooding the banks, all right? So the flow rate needs to be constant. So here's why we're talking about that. The question I want to ask you is looking at capillaries. So actually before that, here, let's talk about this as well. So the flow rate itself needs to be constant. The next thing then is, well, what about the velocity? What about the speed? Here, at this point in the river, if you were to watch like a little paper boat or something, then what you would find is that the flow rate itself is, let's say, reasonably fast. Not the flow rate, the velocity itself is reasonably fast. On the other hand, if you were to look at then the velocity of the water here, then you can imagine that it's going to be meandering a little bit more slowly. And if you've ever gone down and like played by the creek and you've seen this kind of thing, you've seen water rushing through at this point, and then it kind of slowly meanders, and then as the thing narrows, water rushes through again because it's trying to maintain a constant flow rate. So here's again, let's now go into physiology. Why are we talking about that? As blood is moving through the capillaries, you need to ask yourself, do you want the velocity to be really, really, really fast or quite slow? One more time. As blood is moving through the capillaries, do you want that blood to sprint through real fast, or do you want it to kind of slowly meander? And I'm going to tell you the answer should be you want it to slowly meander. And the reason for that is because you want this thing to slowly meander so that there's plenty of time for you to dump off the good stuff and pick up the waste from these capillaries. If the blood were to sprint through, then by the time that blood gets over here, you may not have had enough opportunity to dump off the good stuff and pick up the waste. So it makes sense that you want this thing to slowly meander. But here's the next question. This is interesting. Well, where did water slowly meander? When the pipe was really wide or when the pipe was narrow? If you pause the video, you'd say, well, I think we said that it would slowly meander when the pipe was really wide compared to when it's very, very narrow. So what gives? How does that make sense? Because capillaries themselves are really, really narrow. Remember when you go to this blood vessel kind of comparison table, the capillaries themselves are actually quite small. The diameter is quite small compared to all the other blood vessels. Remember that oftentimes in capillaries, red blood cells like this are going through single file because capillaries are so narrow. So if they're so narrow, why is blood moving so slowly? And here's your answer. The answer lies here. Even though each individual capillary is narrow, and that's where my cursor is running right now, even though each individual capillary is narrow, you've got tons and tons and tons of them. Therefore, as a collective, there's a lot more cross-sectional area. look, here on the x-axis, you've got the aorta. You got one aorta, and then you got a whole bunch of arteries, but even more arterioles and even more capillaries all over the place. Because these things branch off almost like a tree. Like there's a big trunk, which is the aorta, and then there's a handful of slightly smaller branches, and those are the arteries. And each of those branches gives rise to even smaller branches, and those are arterioles. And it continues on into capillaries. And so what I want you to see is that even though the aorta is real big and wide, you only got one of them. So the total cross-sectional area of the aorta is actually quite small. But even though capillaries are real small, you've got so many of them all over the place that the total cross-sectional area of the capillary beds is actually quite large. Therefore, velocity of blood flow is fastest through the aorta, and the blood flow velocity, the speed, diminishes as we get to the capillaries, which is where it bottoms out. Again, this makes sense because you want slow blood flow so that you have plenty of time for exchange to occur. Okay, so where we're at, we described the arterial and venous end of the capillary, and we answered the second learning objective as well. Let's talk about blood pressure now. When you look at blood pressure, I need you to know what kind of standard, typical blood pressure is. You've probably heard that blood pressure is 120 over 80. You've probably heard that. Well, what does that mean? 120 is the systolic measurement, and 80 is the diastolic measurement. And all that means is that if you were to measure blood pressure in the pipes, during ventricular systole, it would be at 120. If you measured blood pressure in the pipes at ventricular diastole, it would be 80. Now, where is this measure taken? Traditionally, it's taken at the brachial artery, which is right in your brachium, right in your arm. So this is your standard traditional number at the brachial artery. So systolic, diastolic numbers. So what I want you to see is that those numbers, if you were to just say, okay, what's blood pressure at the brachial artery right now? Then you'd say, right now I might be kind of at the tail end of systole, or at the beginning or middle or tail end of diastole, and pressure's always going up and pressure's going down. And so your response might be, okay, just tell me the average. Just tell me what the average is doing. This is where averages become useful, right? We like average numbers. And so what I want you to see here is that we have an average blood pressure. What you're looking at here is at the aorta, the systolic blood pressure is 120, the diastolic pressure is 80, okay? And so then, and look, I know I said at the brachial artery. Well, the reality is it's slightly higher at the aorta than it is at the brachial artery because you still need a pressure gradient to advance the movement of blood. It's just not a hugely dramatic pressure gradient at that point. So with that said, systolic pressure is 120, diastolic pressure is 80. And so we just want an average number. So how do you get the average? You don't just add the two up. 120 plus 80 is 200 divided by 2 is 100. We don't do that. Instead, we've got an equation for it. And we have a term for it. It's called mean, which is the average arterial pressure. What is mean arterial pressure, MAP? Well, here's the equation for it. Mean arterial pressure is diastolic pressure plus 1/3 of the systolic pressure minus diastolic pressure. So if we run the math through, pause the video if it's too fast, but if we run the math through, diastolic pressure is 80 millimeters of mercury, 1/3, 120 is systolic pressure, 80 is diastolic pressure, 120 minus 80 is 40. Remember that we're going to do what's inside the parentheses first. And so then 1/3 of 40 is 13.33, 80 plus 13.33 is about 93. So what is mean arterial pressure? Average pressure is 93. Average pressure is 93, right over here. Okay, and again, this is at the aorta and at most systemic arteries. So again, that is a useful measure for us. Now, here's something for you to think about. I'm going to ask the question. I'll ask it twice, then you pause the video and you think about it. When you look at mean arterial pressure is strongly biased toward what? Diastolic pressure or systolic pressure. Those are the only two options. Is your mean arterial pressure biased toward diastolic pressure, or is it biased toward systolic pressure? If that language sounds weird to you, let me say it this way. The mean arterial pressure is what is a greater contribution to mean arterial pressure? Diastolic or systolic? And when you look at the equation, you'd say, well, systolic minus diastolic is 40, but the base of it seems to be diastolic pressure. And you're right. So it's biased towards diastolic pressure. Well, why on earth is this thing biased towards diastolic pressure? Well, your answer lies here in your other PowerPoint. It's in this time in the cardiac cycle. Remember that our heart spends most of its time in diastole. If you look at this, look, if we were, I don't need you to memorize this timeline here, but if you look at this, how long do I spend in systole? I spend, let's see, like 0.3 seconds, right? 0.4 minus 0.1 is 0.3 seconds. That's how long I spend in systole. How long do I spend in diastole? 1, 2, 3, 4, 5. 0.5 seconds. I spend half a second in diastole and only 0.3 second in systole. So most of my time is spent in diastole. So because of that, because we're spending most of our time in diastole really is the greater contributor to average blood pressure. And so that's this number. I do need you to know this equation and this number. All right, so that's mean arterial pressure. If I ask you what is the mean arterial pressure down at the capillaries, you'd say, well, it's pretty darn low. And the other question is, what happens to mean arterial pressure when you go from capillaries back into the veins? Most people want to say, well, I think mean arterial pressure goes up. It's got to be lowest at the capillaries, right? Because blood flow is so slow, I would assume pressure is the lowest at the capillaries. And I can see why you'd want to say that, but how about this? What does blood need in order to move? It needs a pressure gradient, right? We know that. It needs a pressure gradient. And so if pressure is higher in the veins than it is in the capillaries, how is blood going to move forward? And the answer is it's not going to. So what we see is that the mean pressure, which is this line over here, that mean pressure continues to go down all the way through the vena cava and eventually will dump into the right atrium, and it cycles through all over again. This mean arterial, you need to have this constant gradient hill, this hill right over here. If mean pressure was over here, if it swooped up like I'm doing right now, if it swoops up, right, like this, then obviously you don't have a pressure gradient and you're not going to be advance blood. You're not going to be able to advance blood through the venous system. Okay, so there's mean arterial pressure. All right, now let's get to the last part of this this video, and what I want to talk about now are the forces that drive exchange at the capillary. Remember, at the arterial end of the capillary, I dump off the good stuff, and actually, I guess I didn't tell you, but you pick up the waste at the venous end of the capillary. All right, so exchange occurs at the capillary. So I want you to picture this, how it goes from red to blue as I deoxygenate my capillaries. What I have here are the different forces. Now, what I want you to see is that this right here, this thing this big tube is your capillary, and it's bright red over here, and it becomes less and less and less and less and less red until it becomes blue. Everything here in red is the arterial end of the capillary. Everything here in blue is the venous end of the capillary. And what you're seeing here is exchange occurring. Now, what I want you to see is what are the forces that drive exchange? Here it is. You have forces that drive things out of the capillary, and that force is hydrostatic pressure. What is hydrostatic pressure? Honestly, it's just mean arterial pressure. It's just blood pressure. Okay? That drives things out of the capillary. When things move out of the capillary, we call that filtration. But on the other hand, there are also forces that pull things into the capillary. And what is that force? It's colloid osmotic pressure. It's this thing called colloid osmotic pressure. What on earth is colloid osmotic pressure? Well, you know what colloid osmotic pressure is. You've seen things like this before. Look, here I got chamber A filled with water and solutes. Here I've got chamber B filled with water and solutes. Let's just pretend this is a selectively permeable membrane and these big yellow solutes cannot move. Water's going to move, isn't it? That's what we call osmosis. So which way does water move? Does water move from chamber A into chamber B, or does water move from chamber B into chamber A? Well, you should know the answer to that. Water is going to move from chamber B into chamber A, because that's what osmosis is. Osmosis is the movement of water from areas of low solute concentration to high solute concentration. What drives that? It's osmotic pressure, or colloid osmotic pressure. So that osmotic gradient is set up such that water wants to move from chamber B to chamber A. Now what I want you to imagine is, I want you to imagine taking this whole thing and rotating it in this direction. Okay, if you were to take this whole box and kind of spin it around, then this chamber A over here, and this is chamber B, what are we going to see? Then the pipe right over here is actually chamber A. Here, let me steal this letter and drag it in here. This is chamber A, and then chamber B is going to be this thing right over here. Okay, so this is chamber A and this is chamber B. Which way does water move? Well, water, here, let me take some of these dots here. Water is driven by all these tiny little solutes, and I know that's huge. Here, let me try to make it a little bit smaller for you right over here. So I got all sorts of these little dots all over the place, and that's going to drive water in. So that's this blue force, colloid osmotic pressure. When stuff moves that way, we refer to that as reabsorption. All right. So now, what are these yellow dots? You know. You've already learned blood. It's albumin. It's albumin and other types of plasma proteins. So those things contribute to colloid osmotic pressure, and that drives this reabsorptive force. So now with that said here, let me make these things disappear, because hopefully you have that sense now. I'm going to replace my letters here. And so now here's my question for you. At point A, what two forces are present? Well, you've got hydrostatic pressure in red driving things out, colloid osmotic pressure and over here also pulling things in. Who's stronger? Well, this one is bigger. It's a bigger, fatter arrow, so hydrostatic pressure is stronger. What ends up happening then, as you travel from point A all the way to point G, what happens to blood pressure? What happens to hydrostatic pressure? It goes down, right? Because you need a pressure gradient. So what I want you to see is this arrow subsequently gets smaller and shorter and skinnier, indicating that hydrostatic pressure itself diminishes throughout the length of the capillary. So by the time you get to G, it's actually a lower hydrostatic pressure. Hang on, did we see that? Yep. At the capillary, beginning of the capillary towards the end of the capillary, hydrostatic pressure will go down, which is blood pressure in this case. So great. What about colloid osmotic pressure? If you look at these blue arrows, they're actually all the same length and thickness, because colloid osmotic pressure does not change from point A to point G. What ends up happening, though, is at point A, who's stronger? Hydrostatic pressure. At point B, who's stronger? It's still hydrostatic pressure. At point C, it's still hydrostatic pressure. At point D, it's evening out, but it looks like it's still hydrostatic pressure. So from A all the way to D, what we're going to see is filtration. From E, who's stronger? Ooh, now it looks like colloid osmotic pressure is stronger than hydrostatic pressure. So now there's net reabsorption. What about at F and G? Same thing. Colloid osmotic pressure did not change from point A to point G. It's still there, but now it is stronger than hydrostatic pressure, which did change from point A to point G. And so therefore we see net reabsorption here at the venous end of the capillary. All right, so if you want some numbers, there are some numbers. I don't need you to memorize these numbers, but this is the same thing, right? It's the same pipe, just shown in a different artist. This is me in PowerPoint, and this is from another textbook. So this is the arterial end of the capillary and the venous end of the capillary. HP is hydrostatic pressure. The hydrostatic pressure is 35 at the arterial end of the capillary. At the venous end of the capillary, it is 17. So this would be 35 over here and 17 over here. So really what that means is 35, 34, 38, 37, 36, 35, 34, 33, 32, 31, as I'm going from A to G, so on and so forth, until you get down to G, it would be 17. 25, though, is colloid osmotic pressure, and you see at the arterial and the venous end, it doesn't change. It's exactly the same, and that's driving reabsorption. That arrow comes into the capillary, whereas hydrostatic pressure, that pushes things out of the capillary. Now, if you do the math, you'll see 35 minus 25 is net filtration pressure. That's what NFP is. It's net filtration pressure of 10 millimeters of mercury out. Here, they're saying net filtration pressure of negative 8 millimeters. All that really means, and so I don't need you to care about these negative numbers here. Again, you don't have to memorize the numbers. It just means the colloid osmotic pressure wins. That's that net OP is net osmotic pressure. It wins. The negative number came from here, okay? And that's pulling stuff into the capillary. The net effect is what? You dump off the good stuff at the arterial end. You pick up the waste at the venous end. And these are the forces driving that. All right, last two slides are nothing new. This slide, I'm not going to go through it, but pause the video if you like and spend some time just staring at that slide. It's the exact same thing. Look, some of the numbers are going to be a little bit different, but potato, who cares? The theme is the same. Okay, so there's net filtration at the arterial end of the capillary and net reabsorption at the venous end of the capillary, with the two opposing forces, meaning our hydrostatic pressure, BHP here is blood hydrostatic pressure, and then colloid osmotic pressure as the other one. Okay, so they throw in a couple extra pressures over here. Don't worry about it, not a big deal. This and this is what I need you to know. This slide, I'm not going to spend any time on it at all, is simply to say that there is control over blood vessels at large, okay, in the big picture. And this, it almost seems a little bit misplaced since we're really talking about capillaries here, but as long as I have it, this is, let's say, some arteriole, and it's telling you that there are different variables here that are going to make a blood vessel vasoconstrict or vasodilate. And these are also like oxygen levels, CO2 levels, pH, potassium levels, adenosine, nitric oxide, ANP, ADH, catecholamines, whatever. These are all going to have different effects on blood vessel diameter. So what that really means is, look, blood vessel diameter, as you recall, is really the major contributor to blood pressure regulation. And so it means that there's a whole bunch of stuff out there in your body that can be involved in helping to regulate blood pressure. All these chemicals can be released. Okay, so that's really the big picture. I don't need you to know the specific names of any of these, though. All right, to recap, we've done number 1 and 2 already. We described blood pressure, systolic, diastolic, right, 120 and 80, and mean arterial pressure as well, too, strongly biased towards diastolic pressure. We described the forces that promote exchange at the arterial end as well as the venous end, and those were hydrostatic pressure and cholerismatic pressure. You also need to know the terms filtration and reabsorption. That's not on your learning objectives specifically. However, it should be, because I kept using those terms here to describe filtration and reabsorption. So please make sure you know those terms. And that will wrap it up.